Welcome to the digital lab tour about the electron microscopes and focused ion beams, which are accessible at the Institute of Materials Physics. My name is Tobias Meyer and I'm responsible for the instruments that I'm going to present. Let's first start with the scanning electron microscopes as well as focused ion beams. We have two dual machines, the Thermo Fisher Helios G4 as well as a nanolab, which are both scanning electron microscopes as well as focused ion beams, which can, for example, be used in order to prepare TEM lamella, which are then later on analyzed in the transmission electron microscope. But these instruments are way more versatile. You can not simply prepare lamella, but you can also use them for imaging, of course, as well, and also some rather advanced techniques, which I'm going to mention later on. We also have one scanning electron microscope without a focused ion beam, but with the capability to tune the pressure in a certain range up to the millibar range, which is then called a variable pressure SEM. And that technique can be used to reduce charging, for example, of specimens, and also in order to study rather wet materials as well as some reactions to low pressure gaseous environments. All of these instruments are equipped with a field emission gun as well as piezo stages, which means that we can rather decently position our samples and have quite a good imaging capability. We have a lot of different detectors spread over these instruments. I would say all of them are, of course, equipped with the standard SEM detectors for secondary electron detection, but we have also the capability to collect backscattered electrons, which can yield very nice crystalline contrasts. For example, if you have multicrystalline materials, you can map the domain structure. We also have the capability to go to STEM conditions in these two instruments. We have the ability to do electron backscatter diffraction in the variable pressure SEM, where you can really map the orientation and not just differentiate between different crystallites, but you can really pin down the exact crystalline orientation at the surface. We also have the ability to do EDX in the variable pressure SEM, and we also have different opportunities to go to in situ experiments, like, for example, cooling down the specimens. We have also micro manipulators that are shown right here, where you can mechanically as well as electronically contact your samples, and actually many things more. And I'm going to present our most recent microscope, the Helios G4, in the video shown on the next slide. We are now entering the lab of the Helios G4, which you can see here in an overview in the beginning. We have the chamber door at the front, where you can insert larger specimens, for example. We have the electron gun coming here from the top, where our electrons are extracted and accelerated. And we have the ion gun coming from the side, so we have also two different perspectives to our specimen. And the electron beam is typically used for imaging, whereas the ion beam can be used for structuring of the sample. And in combination, you can, for example, extract nanostructures and transfer them also to different places by using a micro manipulator. That's the guy here in the front. It's a very sharp needle that can be inserted and moved by piezo-controlled engines, which means that you can also position it very precisely and handle rather delicate small specimens. If we go to the side, we see first of all the ion gun a little bit better, and also that we have different gas injection systems where we can inject the gas inside the chamber, for example, a carbon platinum alloy, and that can be used in order to deposit in the end some materials on the specimens, because the ion beam as well as the electron beam can crack the gas, and this way we can locally deposit materials like, for example, a carbon-platinum alloy on top of the specimen, which can, for example, be used as a protection bar for TEM lamellar preparation. If we now move a little bit further to the side, we see that we also have another needle which can be inserted in the chamber. That's a micropolisher combined with a gas inlet which contains argon, and this can be used in order to ignite a local plasma with our electron beam inside the chamber. That's a rather recent development, and yeah, this way you can really locally use a plasma in order to clean the surface of your sample. If we look at the back side, we see that there is the connection of the high tension, for example, to the electron gun, and also parts of the vacuum system like the IGPs, which are differentially pumping this electron gun to increase the vacuum inside the gun compared to the vacuum inside the chamber. And if we now go to the other side of the instrument, we also see that the FIP is equipped with a load lock. This guy can be used in order to very quickly insert and remove specimens to and from the chamber, which saves you quite a lot of time and is quite convenient if you, for example, just want to take a quick image of a freshly prepared sample. Then you get it within minutes. Yeah, that was the overview of the G4. This is our most recent scanning electron microscope, dual machine with an FIB, and it's really a working horse, which helps us quite a lot in order to create nice specimens and also to get very nice images very quickly. Yeah, another fraction of our electron microscopes are of course the transmission electron microscopes, which are shown here. We got 4 of them at the moment. Firstly, of course, to mention our FEI Titan ETEM, which is an environmental TEM, so we can also let in gases, and I'm going to show that a bit more in detail on the next slide. But we also have rather older machines, and I would like to emphasize them here as well, because of course the most recent machine is a bit more complicated sometimes to be operated, but an introduction and training on one of these older machines, for example, which are perfectly fine to get a quick feedback, for example, for a growth department, is rather easy. So people working with you could also autonomously do TEM experiments, for example, on the CM12, which is pretty easily accessible, and get a short notice feedback on how well growth has succeeded or not. To get some specifications of our TEM part, we have the ability to resolve 1 angstrom structures in TEM mode in our Titan, as well as 1.4 angstrom in STEM mode. We have a lot of in situ capabilities. Firstly, of course, to mention the gaseous environment that can be used in the ETEM, but we also have cooling stage. We have a heating and biasing stage. I've put that in one point because you can do that simultaneously. We also have acquired a liquid cell, so we can study materials inside a liquid. In the TEM, we have a straining holder for mechanical testing, and we are quite versatile in different TEM methods, I would say. We have analytical methods like EDX and monochromated yields, so we can also go to high energy resolution in yields, and we also are able to do advanced methods like 4-dimensional scanning transmission electron microscopy, as well as off-axis electron holography. We're now entering the ETEM lab, and as you can see, this instrument is rather tall, so we need high ceilings in the lab. And I'm going to talk about the interior of the microscope itself later on, but first of all, I would like to emphasize our gas inlet system, which makes the instrument a bit more special than a regular Titan. You can see that from the floor we have different gas tubes of helium, oxygen, argon, nitrogen, carbon dioxide and monoxide, as well as hydrogen that we can use. And we can also create gas mixtures by using these mass flow controllers and letting those gas mixtures into the octagon, into the column of the microscope. One more gas, which we use as a vapor, is also accessible. That's water. And you can see that there is a small tube here where we have liquid water, and the vapor that's created in low pressures of that liquid vapor water can then also be used in order to put it in the column of the microscope. You can see here the gas inlets. If we now go to the microscope itself, you see it's all covered behind plastic, but we can open this cover and have a look at the interior. We will see that we have a field emission gun here at the top. It's pretty large because there is also a monochromator included, which we can then use for this high energy resolution in yields. We have a 3 condenser system, which makes these advanced methods like 4D STEM available. We have the sample port here at the side. That's where the specimen goes into the column. And yeah, here you can also see some tubes which are related to the gas system. So this is also where locally the gas is going to be injected into the column, and then we have again differential pumping in order to maintain a high vacuum up here at the electron gun. We have a corrector, an image corrector, to correct the spherical aberration of the objective lens of the TEM. And that's basically the reason why the TEM resolution is much better than the STEM resolution, because it is designed to be advantageous in the TEM mode. Otherwise, it had to be on top of the sample, because in STEM we focus the probe on the sample. Yeah, if we continue the video, then we will see we have also the biprism here at the very lower part of the microscope, which can be used for off-axis electron holography. And yeah, soon I'm going to remove the cover of the flue screen to show that there are actually electrons reaching the very bottom of the microscope. So that's how we operate. And if you then lift the screen, you can see that by using digital cameras, we can map our specimens and move them along as we want to. Yeah, on the side we also have the EDX detector being attached to the column for chemical analysis, and we also have below the instrument, that's not so well visible in this video here, we also have an image filter which can be used for both energy filter TM as well as also the yields methods that I was mentioning before. And lastly, I would also like to mention some TM sample preparation techniques that we got. Of course, these instruments look less impressive, let's say, than the tall guys, but it's also quite important to have high quality TEM specimens in order to get nice data in the end. And yeah, we also got quite a versatile collection of preparation tools in order to pursue conventional preparation instead of the focused ion beam preparation that was mentioned in the beginning. And yeah, definitely to be mentioned are the 2 precision ion polishing systems shown here, which we can use in order to drill holes with a low energy argon beam, where we can go down to a few hundred electron volts acceleration voltage in order to reduce the beam damage of the ion beam and end up with nicer qualities and nicer surfaces of the prepared samples for the TEM. We have also many opportunities to make larger structures in the beginning a bit smaller in order to fit to our TEM holders. So we have grinding machines, a diamond saw, and we can also use electropolishing, for example, which works best for metals in order to make the electron transparency being possible just by polishing the sample electrochemically. We also have a dimple grinder as well as a tripod polisher, and of course also people who have quite some experience in how to use all this equipment. Yeah, generally, maybe as a final remark, yeah, I hope you enjoyed the Electron microscopes lab tour, and yeah, I'm very happy to get to know you. I'm going to be part of the lab tour team as well, so I hope to meet you there, and yeah, to start a fruitful collaboration between very nice sample growth as well as sample characterization in our electron microscopes.